And for speaker, I am telling, sir, we have various people from the various fields, space technology, then optoelectronic materials, then someone about e-mobility, e-vehicles, and one person from industry, he came and he talked about that instrument details about different characterizations. And we have planned something, but it was excellent. What we have think, it is more than that. It was very happy. So today also we are very eager to hear from you, sir, because as a doctor, you are presenting about some ophthalmic materials and something about the retinal function, and these are very new thing for me. As a teachers, as a faculty members, definitely it is going to be very new. I believe we have nearly about 170 to 180 people on our ATAL FTP every day. Maybe no one has heard about these things, at least to be in the recent past there in service. Maybe in school days we have heard about that retina and etc. So now definitely it is going to be an eye opener and good session for us. With this expectation, I welcome all the participants and especially I welcome and I thank our presenter today's research person, Dr. Saravana Kulandevilu sir, and is brother of our friend and our colleague Swaminathan sir. It was very great. We are having two persons on this screen. And now I request Dr. Tangarasu from Kalasalingam University. I request Tangarasu to give a brief introduction about our speaker. Then the speaker will take over this.
Good evening, everyone. I
Good
am
evening.
pleased to introduce our distinguished resource person for today's session of the Faculty Development Program. We are fortunate with Saravanan Kulandevalu sir. He is working as an associate professor in West Virginia University and in the Department of Ophthalmology and Visual Sciences. Sir completed his PhD in the year of 2004 from All India Institute of Medical Sciences. Sir published a good number of research papers and he received many travel grants. With this introduction, I welcome you, sir. Now I hand over the session to Dr. Kulandevalu, sir.
Thank you, Dr. Adimulam, Tangaraju, and Swaminathan, and everyone. Thank you for your invitation, and it's for me. It's a pleasure to present what my lab is working on, what I learned like last 25, uh, uh, 30 years of my journey in research. Being from a small village to now the current position. Let me introduce myself. I'm like whatever Dr. Tangaraju talking about myself. I'm Saravanan Kulandevalu, working as an associate professor here. And I'm going to talk about the mechanism associated with the vision. We all see, we all enjoy nature, friends, family. So how this visual response is taking place? What will happen if there is an issue with the vision, like the devastating condition, like we don't see anything, we don't know anyone. This is what I'm going to talk about. And uh, what you are seeing in the screen, first picture, this has been made in 1900, almost like 1906, like when we don't have the technology what we are having right now. And the scientist from Spanish visualizing from the patient how the neuron connected with each other and sending signal to one from another and propagating their signal and to see the response, the light vision. So that's what the picture which you are seeing. So this is going to be really different topic than what you are doing. It's a completely it's a science, and also now use something like molecular machines, motors, like how it's associated with the vision. That's what I'm going to talk about. And before that, and I'm going to disclose, there is no actual conflict of interest relation to this program or presentation. And so I will give a brief introduction about outline, and I'll talk about some introduction and the project which we are doing and summarizing those projects. And three missions, the molecular motors which I'm going to talk about today. One is these are like molecular motors you can call as a proteins, and one is that PRCD and electric impulse, the maintenance of electric impulse in the cells, like it's a ATH, sodium potassium ATPase, and third one is NMN81. So these three topic I'm going to cover, and I will try to finish within the time limit you are giving me. So these are the missions which I was, and I'm going to give a brief introduction about retinal structure. The vision, like now what we all see, is anyone knows about how the vision works? So I'm going to tell you something about, like now you can see in front of you, the eye is there. This is the eye. Just point drops and I'll open it. Yes, yeah. So the vision goes through from this direction, like when through the cornea, this portion, and the lens, and it passes through the inside of your eye. And when it goes inside to the eye, this is where the visual information pass through the eye, like through the cornea and the lens, and goes inside to the eye and hit back where the soft tissue, like this is the soft tissue called retina. This is the place where the information hit, the light signal hit, and the signal uh, captured by the small, like you can see many cells are here, and these cells are interconnected each other. So it goes all the way here and captured by these cells. It's called photoreceptor cell, and after converting this light into electrical signal, send downstream cells, then another cell, then it collect through the optic nerve, goes to the brain. That's where the visual processing is happen. So I'm going to tell you, one by one, these cells, how does it work? And if, as I said, the photoreceptor cell, in the retina, the first cell which capture the light signal is photoreceptor. There are two types of photoreceptor cells. One is rod, it's like you can see the shape itself, rod-like shape, and another one is cones. And the rod, its function in the dim light condition, and cone function in the bright light condition. We will come back later what is dim and what is bright light. And you see these cells, it's compartmentalized. It's a segmented, outer segment, inner segment, nucleus, and synapse. This is the place where the vision get captured and sent to this portion called synapse. Synapse means the communication between one cell to another cell, and send signal one to another. Then like that it goes all the way to the brain. And you see, when you touch somewhere, immediately you hear, okay, something's touching, or somebody's pinching, that it's something happened to my body. So how do we get within fraction of second? So that's what the communication, how that neuronal communications happen, one cell to another cell, it goes on. The brain understand about those function and those response. And like vision, these cells function like that. And if you look at this, as I said, that outer segment, inner segment, and synapse. And outer segment, when the light goes into from here, from retina, as I mentioned the previous slide, all the way to here, it captured in this place. And when it captured, the signal get converted into electrical signal and sent to this place to downstream cell. So I mentioned before that it function in dim light and bright light. So when you walk over the bright sunny day, when you're entering into home or in shadow place, suddenly you see some changes in your vision. You cannot see properly. It takes 1 or 2 minutes. Why? That during that dim light condition, this cell, the rod cell function, And when there is a bright light condition, the cone cell get function. Like, this, you can see the shape itself, rod and cone. So the rod function in dim light condition, the cone function is bright light condition. And how does this light response? It's captured in the outer segment. Like, if you see this outer segment, so this is like, unlike other neuronal cells, you know that the neuron cannot multiply, and once it made, and that's it. Like, it's die means it's die. So that's how, like, when neuron loss leading to, like, many like, brain behavioral dysfunction. For example, like, loss of memory. Like, when we get old and we lose our memory, happens like in a neuron death. But this cell, photoreceptor cell, like where you are seeing, it's continuous process, like, phagocytosis and renewal. Like, these things happen entire our life, and from our birth till our death, like, this process needs to be regulated properly. Like, 1/10 of the disk in the apex, in the top layer, it's phagocytosized every single day. At the same time, the new disk formed in the bottom. Like, this is how this outer segment, like, this portion of the cell, it's developed in everyday process. So what is there in this cell, and how does it important for, like, our visual process? It is like, the specific place where that machines, like, the capturing lights, capturing machines are present. Who, how they capture the light, that's where we were going to say the next one. There's another molecule called opsin, and this is the, like, major, like, protein, the first protein, like, molecule get absorb the light. So this opsin, coupled with another compound called 11-cis chromophore. So when 11-cis chromophore finds with this, and this chromophore, like, in two different positions, it is binds with the opsin. It's a cis, like, closed conformation, and trans, like, open conformation. The closed conformation means when this option is inactive state, when there is no light. Like, what happened, like, during our sleep, like, this is what it happened, because there is no light response. So this closed conformation we see in the light. So this option present in this area, where I said the rods and cones. The rod cells function in dim light condition, cone function in bright light condition. What we see, like, in cone, like, uh, like, compared to rod, only 3%, and the rods are 97%. And these 3% cones enriched in the central portion of your eye, like the retina. There are three types of cones are present. One is small cone, medium cone, large cone. The small cone, like, function in blue light, and medium cone function in green, and large function red light condition. So this is where we visualize, like, the colors. Like, this 3% of the cones makes you to function in bright light condition as well as understanding the colors. But this one, 97% of the cells present, like, functioning in only bright light condition. So if you look at them, the outer segment, and it's contained lots of, like, double membranous disc-like structure, like, it's called disc. And the disc contain this opsin molecule, like, properly organized in this molecule. And this is how you can see there. When it organized, so what happened when light hit? So you wake up and you put the light on. Like, when you put the light on, like, the dark, you see like this. When you put lights on, this chromophore, like, opens up. It goes into transposition. When it transposition, this activate this protein. Like, when it get activate, so what will happen? The activated rhodopsin, like the opsin molecule, activate another molecular motor. It's called transduction complex. So this three complex protein, like, alpha, beta, gamma, this 3 complex protein, like, detachment of how it get activated, the activated option, like convert, like, add another phosphate molecule into this alpha one and convert into, like, GDP diphosphate to triphosphate. When it conversion kicks out another 2 molecule and it gets activated. So when it get activated, this activate another protein called PD6, phosphodiesterase. And the phosphodiesterase is the one responsible for, like, the final downstream of conversion into light into electrical signal. The activated transduction alpha GTP complex remove this inhibitory subunit and it get activated. What will happen that activation? It's reduction of cyclic GMP to GMP, this molecule. I know that it's very, like, confusing to you, like, this is how the molecular motor here in vision it works. That's how we see each other. That's how our vision function. So this conversion of cyclic GMP to GMP, like, leading to, like, dark opening state to, like, closed state. In light, in dark, what happened? The in photoreceptor cell, like, this is photoreceptor, like this here, like, cyclic nucleated gated channel, like, there's a channel which gets opens up. When the channels gets opens up, like, ion goes inside. What ion goes inside? Sodium and calcium goes inside. The sodium and calcium, when it goes inside, the cell get current, like, depolarized. Depolarization means the current will be minus 40 millivolt. And, like, when it goes inside, the sodium and calcium goes inside, and like, this needs to be stopped. How does it needs to be stopped, and how it goes inside? When there is a more cyclic GMP, this get opens up, sodium goes in. When cyclic GMP levels are reduced, like, as I showed here, cyclic GMP to GMP, and like, this reduced to GMP. This GMP, it closed this channel, so entering sodium inside to the cell, it stopped. That's how, like, our vision work. Now that conversion into light into electrical signal, this signal goes all the way to down, and here where it's transmitted to another cell, and it goes to another cell, ultimately to brain, and final processing. Okay. Now I'm going to talk about how does this transmission, like, to downstream cell, how this polarization, the current change in the second topic I'm going to give. But before that, I'll give a little more about what we have done with understanding about outer segment and structure function. This is, like, now I'm going to use one molecule called PRCD. It's called progressive rod cone degeneration. Like, just forget about the name. It's called PRCD molecule. How it's associated for maintaining the structure. If you don't have, as I mentioned, proper outer segment, it leads to severe defect. And defect means the outer segment, this outer segment portion will be destroyed. When it get destroyed, it's leading to loss of our vision. Like, it become blinding disease. So you can say, like, there are multiple blinding conditions. We say the children born with blindness. Born with blindness means, like, they born with a mutation, genetic defect, and they don't even see their parents, and they don't see the world, nature, everything. And that's what it happened. Like, it's called severe blindness. It's a term, like, LCA, but don't worry, it's a children born with blinding disease. Another one, so we just see, like, vision is going okay, but suddenly there's a change. Like, there's a progression of loss of vision. That is called retinitis pigmentosa. Like, it starts from any day after normal vision to deteriorating, that the loss of vision. Then later, age-related macular degeneration. During aging, we lose vision. So these are the major, like, type of blinding disease what we see. I'm going to talk about this molecule, and this molecule mutation. affect humans as well as many dog breeds. Almost 40 different dog breeds are associated with this protein. Uh, uh, the mutation, leading to slow and progressive loss of this disease. Its affect, you see that the outer segment which I was talking about, the disc membrane where it get present, it's affect there, and when it get affect, and you see how this outer segment looks like. This leads to defect in vision. So it's called retinitis pigmentosa. Retinitis pigmentosa, it is 1 in 4,000 humans. We all having this disease. Over 50 different genes, nearly right now it's a 90 plus genes are associated with this disease, and it can be dominant, recessive, and X-linked. That's the forget about the dominant means recessive is very homozygous. It's a way they can lose. The dominant means still the progression of disease. The X-linked is it's X chromosome associated defect. It's found only in male child. So that's what it is. It's called retinitis pigmentosa, and the mutation in this protein are linked with the defect you see in normal fundus, like our eye fundus. If you go to eye exam and they take a picture to see how the retina looks like, the capillary, everything, this is what it's normal. But the defective one, you see the molecule, the black picules, it's a epithelial cell, pigment epithelium, are enriched in the fundus, and this is the progression of losing the vision. So what will happen, the losing vision, like you lose the periphery of your vision. When you see, you'll see only the center portion, not the periphery portion. This is how you see this. When it get progress, ultimately you lose everything, become legally blind. That's what it happened. um, and so this outer segment, the loss of this protein leads to defect in this, this structure. This is how it is. The person which I'm showing here, this part of work done by Sri as well as Joe. And uh, so as I mentioned, that many mutation in this gene, this is very small protein, small molecule. It's a 54 amino acid. You all know about amino acid, what it is. The gene, like it's like transcribed and translated and made the protein. It's a protein is made up of amino acid. The protein is the one, it's a functional molecule, what we have everyday process. So that's what, this protein is 54 amino acid long protein, have many mutation. You see, 5-6 different mutation. Among these, this is most common mutation seen in human as well as in dog breeds. I said that 40 different dog breeds are having this mutation. Why so special about this mutation? We will talk about it. And this small protein binds with a bigger protein, as I mentioned, that rhodopsin, like the conversion of light into electrical signal, and it's a binding with them. It makes that outer segment formation and maintenance. And this is where the maintaining this, it's happened when you don't have PRCD. Making this portion needed protein and lipids, which aggregated here, you see here. This is what it happened. And when the progression happened, this is normal condition, that outer segment. You see in abnormal condition how the defect is leading to uh, like this kind of structure. And when the aging starts, when the mutation in this gene leads to 30 days, small, it's a progressive loss of outer segment. 60 days you see, and 120 days, you see how severely it's affected. So all these studies has been done in mouse model. Like now we make mouse model, like to study the protein molecule which linked with the disease, how the molecular motor associated with the maintaining the visual function. And so the most common protein I was talking about to you, like it's a common mutation. It's a C2Y. The C2Y means like now one of the amino acid called cysteine in the second position, like mutated and changed to tyrosine, another amino acid. This change in amino acid leads to like trafficking of this protein going to outer segment. This is called outer segment. And see, this protein goes to outer segment in normal condition. When there is a mutation happen here, and this protein stuck in the inner segment, inner side, and doesn't go to outer segment where it's supposed to be here. When it's not going to outside, and this portion of the photoreceptor get affected, and when it get affected, which leads to defect. So like also we see there's lots of accumulation, like when it's needed for like making this portion proper function, like lipids and proteins are needed, that's it gets stuck inside. And when it gets stuck inside, and this portion is getting severely affected. And this is what like the fundus picture shows, that lipid accumulation here. And I'm sorry, I'm going fast since we have to cover the time. So overall, this part of molecule, it's linked with um, defect in, yes, making that outer segment which needed for visual response. So any question from this portion, so I can explain like from here before I move to the next part. If you
Now
don't
it's
have, then I get, yeah.
yeah, 1 minute, sir. It's over to the audience, and if you have any clarification or anything, please. We all feel that we entered into a new field, sir. Whatever the
Okay.
things that you explained, but I enjoyed because as a structural person, crystallography person, and you are telling about the GTP, and amino acids, and we all enjoyed.
Okay. Thank you so much. At least you are getting something. The next one is going to be a little different,
Okay.
and this I think it is the current which I'm going to talk about. Current, what is current? How it's related to cell, and this is what I'm going to talk here.
Right.
And this part of work, like carried out by my students, Emily, Boyden, and Jack, and so this is called electric impulse. Like the changing, like you can ask like how does this communication between cell, one cell to another cell, it's happening. It's the same thing. You can think about electric current, like now you put a switch on, like now you see the light on, like now switch off, lights off, like the same, like that's what it happened, like that electron movement, like how does this movement, like the current, it's maintained in this photoreceptor cell, which I'm going to talk here. As I mentioned that in dark and light, there's a sodium. In dark it goes inside because of opening up this channel, like sodium channel. In light it gets closed because there's a high cyclic GMP leads opening and entering, this ion goes inside. And when there's a light, it reduces and it closes. That's how it stops entering to the sodium ion goes inside. What is sodium ion? Like sodium, it's a cation. Like it's a positively charged, and when it get positively charged, like in dark, it is I said minus 40 uh, millivolt, and in light, like it's a hyperpolarized, and here it's a depolarized and it's hyperpolarized. And when there's a sodium goes inside, and the sodium concentration, like it cannot pour all the time to the cell, it needs to be controlled, it needs to be like removed. So we all having, like you say after heavy work, you feel like you're having headache. Oh, I'm tired, and I feel headache. Why do we have all these things? And you also say that, okay, eat banana, you'll be all right. And why do we say, this is because your cell has more sodium inside to the cell. When there is a more sodium inside to the cell, and cell feel little, like you know it's overwhelmed, and it's depolarized. When there is more sodium inside, and it takes more water inside to the cell, it little bit, it's a swelling, all those things, cell not feel happy. And it become ultimately, if you cannot control the sodium concentration, it'll die. So when it die, then that's where, now we have problem. Who's controlling? Now every cycle the sodium goes in, at the same time it needs to be kicked out, and you cannot keep sodium all the time. So how does it get kicked out? There's another enzyme in the downstream, this portion of the photoreceptor cell, it's called inner segment, like sodium potassium ATPase. This is a pump. This pump, using energy, it kicks 3 sodium outside and bring 2 potassium inside. This is what it does. This pump, one another thing I would like to tell you, we eat our energy produced from our food. It's from that, almost 60% of the energy what you take, the calorie, used to buy this enzyme. 60%, can you believe it? 60% of your total energy used to buy this sodium potassium ATPase, this pump. And remaining energy only used the entire, your body. So you see that how important it is, the whole function. Now you receive, your brain sends signal to your body sends signal to brain. So all these things regulated by, how this message is going and coming, and this pump, and how this pump associated with vision. That's what we are going to talk about. And as I mentioned, this pump is in the inner segment of this photoreceptor cell, and it kicks 3 sodium out and bring 2 potassium in. That's what it does. And after, we used with the animal model, mouse model, after making this mouse model, and we tested, by removing this sodium potassium pump, this molecule, by visual function test. It's called electroretinogram. Now when you check the vision, you see in normal condition, the black line which you see normal condition, there's a peak which you can see. This is your photoreceptor functioning shows that the negative peak, and communication happened to downstream cell called positive peak. And you can see these 2 peaks, negative photoreceptor function is all right, and positive communication to other cell is all right. If you remove this pump, what happened? There is no response at all. So meaning that this protein is very, very important for normal function, particularly maintaining the electric current. It's a crucial. That's what we see. And what happened, when you remove, it lose these cells. These cells will lose almost in 16 days in mice. You lose half of the cell. In 30 days, we lose majority of the cell. In normal, you see this, and lacking this pump, you see this much. And 50 days in the model what we created, and we lose entire portion of this cells. So why do we care these things? Because the mutation in this gene linked with the severe, brain defect. Brain defect means Parkinson's disease, Alzheimer's disease, dystonia, the convulsion, what you see, convulsion, the children goes with the, the convulsion, tremor, because of excessive amount of sodium into the neuronal cell leads to this problem. So when there is more sodium goes inside, this neuron doesn't function. When it doesn't function, it's leading to this kind of defect. Until till now, there is no cure for it. There is no, like, we don't have any other system to get rid of the sodium concentration inside to the cell, from the cell to out. So if we have it, that's the remarkable finding. And this identification of this sodium potassium pump got a Nobel Prize in 1992 or 93 by Denmark scientist. And this is the structure where we see the removal. And when we remove, see the normal cell and defective cell, and you see this protein is missing here, and that's what we look at them. There is a this cell communicate to this cell, and this cell communicate to another cell, and goes to brain. And you see, when there is no protein, like this protein, leading to another protein also we lose. So another protein is needed for maintaining the integrity of this inner cell, and that's what it's happened. So so this is very important for both photoreceptor, both rod cells as well as cone cells. In rod cell, like uh, I told you, like, it function in bright light condition. In cone cell, it function, dim light condition. Cone cell function in bright light condition. So when sodium goes inside and it comes here and get rid of here, when you remove this, the sodium concentration, when it goes in, like during dark, we sleep, like during our sleep, like what happened, dark condition. In dark, continuously we are pouring sodium concentration inside. Sodium and calcium goes inside to the cell, and when it going inside to the cell, it continuously, this pump has to remove that sodium concentration. If it doesn't do that, when we wake up, the cell won't be there. And that's what, it's very, very important. As I mentioned, that 60% of the energy used by this enzyme, like, we all think that we are taking rest, and we take only rest for our body, not for eye. The eye constantly function during your sleep. Like, how it function? By removing the sodium inside, from inside to outside. That's what it does. And in cones also we tested. In cone function, you see, when you remove this and you lose the function, it's normal, and this defective one, you see how much defect we see it. And the protein molecule we lose in this, and that I don't want to explain, and more. And but you see in the activation, the visual, rhodopsin activate transduction protein, you see it's reduced. So meaning that when you don't have this protein, the activation cannot happen. When there is no activation, there is no visual response, and that's what we see it. And this is another thing, when you remove the protein, how normal, and see in abnormal how it looks like. And this also shows that when you remove this and see the how much the sodium inside to the cell, this is the sodium concentration. That's what we say. Summarizing this, changing your electric current, alter the polarization. Alter the polarization means when excessive amount of sodium, it's affect the communication between downstream cell. And the downstream cell, when there is no communication and there's no visual response, it's not sending signal to brain. Ultimately, you don't see it. That's what it happened with the current. The third portion, which I would like to take only 10 minutes of this talk, this portion of the talk, it's done by David. And I started with this project by keeping in mind that why do we all getting aging? how this process is happening. Like young kid, then we become adult and middle age, like going to older age. How this process is happening, and there are lots of studies has been shown that there's a NAD, there's another molecule like nicotinamide adenine dinucleotide, declining that NAD level, leading to like aging process. So that NAD is produced by this enzyme, and the mutation in this enzyme, like NMNAT1, this molecule, it associated with severe childhood blinding disease. The children, either they born with blindness, or within a year they lose their vision. So this is what this uh, enzyme does. There is no cure. There's a lots of mutation in this, leading to blindness. That's how we started. Why it's doing, and at the same time, I also interested in understanding about aging process. So this enzyme, there are three different enzymes. 1, 2, 3 are present. So the one which is present in the nucleus, and 2 present in the cytoplasm, and 3 present in the mitochondria in the cell. So here it's needed for maintaining the genomic integrity. So your gene, which is there in the nucleus, chromosome gene. So how that integrity is maintained, this is what it does. So this produce NAD. That NAD maintains genomic integrity. There's another isoform here in the cytoplasm and mitochondria, which helps making more energy, glycolysis. You read about it, and the glucose, ATP, ADP, all those things, it's a making of this energy. This NAD is a crucial process. When there is no NAD, what will happen? The precursor of NAD, start activating the death signal. The death signal means it's allow another signal to kill the cell, destroy the cell. So that's what this protein is very important and major focus right now to the scientific community. And I'm going to talk about this protein. Since this mutation in this protein leads to severe blinding disease, so we made this mutation in model to study, again mouse model. And you see, within 4 days, after born, you see the normal retina, it has become thin. In 10 days, you lose majority of this and majority of this, and you see only this portion. In 30 days, you don't see these two portions, only see this portion. So suggesting that maintaining the structure and function is needed for this enzyme. What exactly this enzyme does? It helps to produce NAD molecule, which is crucial for cellular function. And that's one thing. And how, by removing this enzyme, leading to severe defect of this cell, and what mechanism going on, that's what we study. And when you see here, there's a many cell types, bipolar cell communicated with the photoreceptor, and this cell communicated with the horizontal cell hematocrine cell, and ganglion cell, all getting affected. Like this cell getting affected, and this cell, these three type of cell, it's affected means it die within 10 days. That's what, within 30 days you won't be seeing all these things. And how, like which leading to rapid cell death, it's because of, like there is a problem in differentiation. Like when you have three different layers, this layer, as soon as the kids born, it start getting differentiation. Differentiation means, like you see this cell, like as soon as it born, you can see like this, all cells are like together. Then in 4 days it start differentiating, like this portion, this portion, and this portion. And 10 days you see the photoreceptor is here, bipolar cell, other cell types are here, and this cell is here. So there is a differentiation problem we see here. So NAD or NMN81, it's very important for differentiating the cell. You see in P4, the cell get differentiate, and here when you remove this, it doesn't get differentiate. When you start getting differentiation, mean the maturation of separating, okay, photoreceptor and bipolar cell, leading to severe loss. And that's what we see in this portion. Then, like that is in rod response. So what happened to cones? Like now we remove in cones also the 3% cones, and Sami is working on this portion of the study. When you remove them up, like we lose cone function as well, like in 30 days. And you can look at them, like this is photoreceptor, like rod's function, this is cone function. When you remove in cone, it's a cone response is completely gone. Same time we also start losing that rod response. And the rod response, like as I mentioned, that there's a negative peak, it's a photoreceptor function, and the positive peak is on bipolar function. It's a communication between downstream cell. And what we see, like there is a response here, the rod, but there is no communication between photoreceptor and bipolar cell. That's what we see here. And you can see here clearly there is no communication there. And but it function, the photoreceptor function, but no communication. Why does no communication? The differentiation is affected. So that's what we found from this model. So how does this differentiation affected? Like these two cells communicate here, and when it communicate with the synapse, and you see in normal condition, and like here, and in abnormal condition, like this protein is really localized in the normal condition. In the abnormal condition, you see it's everywhere here. So when it everywhere here, the communication is really getting affected, leading to visual deficit. So overall, this portion of the study, what we talk about, this NMN80 molecule, one molecule, which is responsible for producing NAD, and the mutation leading to severe blinding disease. When we remove, like now, we don't see the survival of the photoreceptor and downstream neuron, and it severely degenerate those neurons. And also, like now, what we see, like I didn't show many data, like why the loss, what happened, how the energy produced, how gene expression changes, like now all these things are affected in this model. So this communication, because of loss of energy, and leading to death of the photoreceptor and bipolar cell. And that's what right now, like three portion of the talk which I was talking to you all. And this majority of this work done by my lab people, and the Sri Emily, Joe did first portion of the talk. Second portion of the talk, Emily, Gabriel, and Joe, uh, uh, Jack, and Biden was doing. And anonymity one mostly done by David, the David here, and also Saminathan. Uh, so this is the my presentation. Hope you understand. But since it's a biology background, I don't know how much you get into it. The funding what we received from our federal grant and the startup as well as other people, this is my lab people, and so who contributed their work, the presentation what I gave to you. So thank you for listening me. Please, any question, I welcome your question, and I can talk to you anything you like.
Thank you,
Thank
Professor. It was
Yeah.
very nice presentation.
Uh,
Right, we got a lot of input about that molecular. I'm very much interested on that, so it was very nice. And I will discuss with your brother Swami about these things, and I will come to you again once again. It was very much interesting to know about the thing. Thank you.